Swayam Prabha. Digital India. Educated India.
Welcome to the lecture number 22. So, till last lecture what we have finished is the instrumentation for the lifetime measurement, and today I would like to start a new topic, that is the effect of intermolecular processes on fluorescence. And basically what we are going to discuss is that how the fluorescence intensity will change because of different intermolecular processes at the excited state of the species. So, the most well known or well discussed phenomena for such kind of process is broadly represented as the fluorescence quenching, and in the fluorescence quenching, let us say you have this fluorophore solution in your cuvette, and upon excitation of suitable light source, right, the molecule will start fluorescing. So, let us say this particular molecule is giving bright green color fluorescence over here. Now, on addition of some outside agent, right, as I am adding over here, you will see that just I am adding one drop of the other molecule in it, and the color is like not that strong fluorescence, right, the fluorescence intensity decrease, right. Again, if I add little more of this compound from the outside, then the fluorescence intensity decrease again, and that will keep on go on as long as we see that concentration dependent fluorescence intensity change of this species. So, this phenomena is known as fluorescence quenching. So, in this case what we are going to see is, if I plot the fluorescence intensity as a function of concentration of that other species, right, whatever I was adding over here. So, if I plot the fluorescence intensity as a function of the concentration of this compound which I am adding, generally known as the quencher molecule. So, I was adding quencher molecule to my solution containing fluorophores. Okay. So, from here what I can do? I can simply plot the fluorescence intensity as a function of concentration of this quencher over here. So, let me do that. So, in that y axis let me plot fluorescence intensity, just simply denoted by I, and the x axis is the concentration of quencher. So, if the fluorescence spectrum is very strong, right. So, intensity is very strong when there is no quencher, that means 0 quencher. So, I will start from this point, and then as I will add this quencher, as the concentration of the quencher will increase in the solution, then the fluorescence intensity will keep on decrease. So, it will start from a value. So, that depends on the excitation light source, the concentration of the fluorophore present in the solution, like these are the main thing, and obviously the quantum yield of the fluorophore. So, depending on that I will get a particular value of intensity, and then that intensity value will keep on decrease like this way as I will change the concentration of the quencher. However, in generally this type of representation was not done in fluorescence quenching experiment. Rather, what people do is just plot I0 by I versus concentration of the that molecule, quencher molecule, right. This quencher is just represented as Q. Okay. So, if I now plot I 0 by I, what is that I 0? I 0 is the intensity 0 means for 0 Q, then that is called the I 0, and then all those others are I for a different value of Q. So, what I am going to get when Q equal to 0, then I 0 by I is equal to I 0 by I 0, this is equal to 1. So, it will start from the value 1. As I am going to increase the concentration of the quencher, the fluorescence intensity will decrease. So, the I 0 by I, this value will be greater than 1. So, that total quantity will be greater than 1. So, what you will going to get? You will get such kind of plot, and with a specific slope, and that slope is equal to k q tau 0. I will come with this derivation later on. So, this kind of value, that whatever I have written, this slope is called the Sternberger quenching constant, which has some significance which we can discuss later on, but this is the whole as a phenomena, right, of the fluorescence quenching. So, what is going on? I am going to add some molecule which is referred as the quencher. Quencher means is quench something, that is the fluorescence. So, by addition of those outside agents, outside molecule to a solution of fluorophore, which is actually giving fluorescence emission from the sample, the fluorescence intensity is quenched. So, fluorescence intensity is becoming lower and lower as we add those type of molecule like the quencher molecules to the solution, and that is best represented in the literature or in several books like I 0 by I. If you want, you can also represent it by just simply plotting I versus Q, but generally it is done for the convenience I 0 by I versus Q, where you will get a straight line and the slope is known as the Sternberger quenching constant. I will come to the derivation of this, as I said. Okay. Now, let me give you some example of the reason for this fluorescence quenching. Now, obviously, we will going to ask that why such fluorescence intensity is decreasing by the addition of the quencher. There could be several reasons, right. First, I have written the collision with heavy atom, right, that could be one of the reason, or collision with the paramagnetic species or electron transfer. So, in this case, let us say for the electron transfer, the fluorophore which you have is actually in the excited state, and you remember that excited state is denoted by putting a star marks after the molecule. So, here I put the molecule as D, and here you see a star marks, that means this molecule D, which is known as the donor, is in the excited state. Now, if some other species, that Q I was talking about, that Q here is A, if A comes and interact with the D star, so that this D star is getting deactivated with some rate constant. So, you see here there is no star, that means this is not in the excited state, that means fluorescence will not come from this species. Eventually, you can also excite the A star A, like the acceptor in the excited state, and this is now your fluorophore. Your fluorophore not necessarily be the donor, it also could be the acceptor. So, this is for the electron transfer, but probably we will not going to discuss this electron transfer process because of this basic nature of this course, but it is just for your information that what are the different process which are responsible for this fluorescence quenching. We will discuss fluorescence quenching in detail, but those processes, right, that is what I am discussing here, maybe will not going to be covered in this course. Okay, so, the other type of mechanism is X-imer formation, like you see here this molecule M is in the excited state, right, this star, look at this star marks, and it may interact with another molecule which is not in the excited state and giving something like this, where the fluorescence of this guy is totally different than the fluorescence of this guy. It means that you will going to see the decrease of the fluorescence intensity for this M star molecule. It also could be the exciplex formation. In this case, this type of molecule, right, is interacting with a different type. Here this is M, this is M, this is in the excited state, this is in the ground state, and this is in the excited state, but here is totally different molecule. Although this is in the excited state, this is also in the ground state, but they are different molecule, and they can also form such kind of complex where the fluorescence signal is quite different than that of A star or D star, and you will see the decrease in the emission intensity of D star or A star. So, this is nothing but another type of fluorescence quenching. We can also have this proton transfer over here. So, probably this one I am going to discuss little bit. So, in this case that if the molecule which you are going to use as a fluorophore, that means which will going to give you the emission under photoexcitation, right, that is my fluorophore, that is my fluorescence, right. In that case, this one of the hydrogen may be transferred to one base to make the conjugate acid and the conjugate base of this molecule. So, in this case the nature of emission of AH star and A minus star, they are quite different, right. So, you will going to see the number of AH star is being reduced because of this proton transfer, and the formation of A minus star is taking place. So, as the number of AH star molecule is decreasing in the solution, so obviously the fluorescence intensity will decrease. So, which is nothing but a kind of quenching, that is the fluorescence intensity will decrease, that means the fluorescence will quench, right, for AH star emission. Similarly, I can talk about this B star. So, this is exactly same thing, and we can also have this energy transfer as a responsible for the fluorescence quenching of a particular band. This one we will going to discuss in detail later on, because this is an interesting topic. Okay, so, let me draw one diagram over here. This is again very simplified Jablonski diagram, as I already discussed, right. So, let me draw the energy level of a molecule under investigation, which is my fluorophore. So, here is my S0 state and here is my S1 state, right. So, if the transition is allowed transition under suitable excitation, I can promote molecules from S0 state to S1 state, that we have already seen, right. And after being excited, so the fate of excitation we are already discussing, that molecule will come back from the excited state to the ground state mainly by two different pathway. One is designated as the radiative pathway, where the dissipation of the excess energy is in terms of light, and the another types of pathway is the non-radiative pathway, where the dissipation of the excess energy is in terms of heat, you remember. So, this is my radiative pathway K R, and I would also have this non-radiative pathway K N R, and I have already defined this fluorescence quantum yield of this molecule as K R by K R plus K N R, and the lifetime of this I have already defined as 1 over K R plus K N R. Now, what I am telling you, that obviously as you have seen that on addition of this Q is actually decreasing its fluorescence intensity, and that decrease of the fluorescence intensity, it depends on the concentration of this quencher molecule. So, there must be another pathway by which there is a deactivation, and this deactivation is type of non-radiative deactivation, because in this case there is no emission. If there is emission of photon, then the fluorescence intensity will not going to be decreased. So, here it is a fluorescence intensity is decreasing, that means this is a kind of a non-radiative process, and it depends on the quencher concentration. So, I write this as Kq into Q. So, this is another pathway by which the molecules are coming back from the excited state to the ground state. So, as these are in absence of that molecule, these are means this expression of the quantum yield, right, fluorescence quantum yield and the expression for the lifetime, as they are in absence of this quencher molecule, I would rather replace this term as phi 0 and tau 0. Right. Now, in presence of this quencher, this another pathway is present, right, k q into q. So, in that case what will happen? If I want to write this phi, then this phi I will write as k r. So, this is only the radiative process by which the photon is emitted, right, and all others are whatever this extra k q into q, this is the non-radiative process. So, I will write here k r plus k n r plus k q into q, and in this case I will also write like that way 1 over k r plus k n r plus k q into q. Okay. Now, what I will do? I will take the ratio of phi 0 by phi, is not it? So, if I take this ratio phi 0 by phi, what I will get is k r plus k n r plus k q into q divided by k r plus k n r. So, this is equal to 1 plus k q into 1 by k r plus k n r into q. So, this is equal to 1 plus k q. What is this? This is nothing but tau 0, you see here tau 0 I have defined in this case, tau 0 into q, and this is this k q into tau 0 is written as k d to q, or somebody also write as a k s v, and this is called the Stern-Volmer quenching constant. Okay. Now, if you do the same thing over here, like if you take tau 0 by tau, then what you will get? You will get exactly the same expression, because here this k r and k r cancels. So, this is nothing but that same expression, right. So, in this case you will also get 1 plus k q tau 0 into q. So, what you have seen, this phi 0 by phi is having such kind of equation, tau 0 by tau is also having such kind of equation, where this quantity is k d, Stern-Volmer quenching constant. Okay. So, as I told you that if you plot now phi 0 by phi, plotting phi 0 by phi and I 0 by I is same. Why so? Because same under the constant excitation, right, under the constant excitation light intensity. Okay. So, this is same as I 0 by I, that what I was telling you just a few minutes back. So, if I plot, this plot will start from 1 and you will get this type of different values as a function of q, right. Now, if you plot tau 0 by tau, if you plot tau 0 by tau instead of phi 0 by phi, see here the slope is exactly same. So, you should get exactly same slope as phi 0 by phi, tau 0 by tau and phi 0 by phi are exactly same, right. So, in this case just try to visualize this thing. There is a fluorophore which is giving fluorescence, it has its own radiative pathway, own non-radiative pathway. Now, there is an external agent which is called the quencher is coming, interacting with the fluorophore, right, because of the collision, right. So, it is interacting and then it is creating a further non-radiative pathway which I have denoted as the Kq, which is also depend on the concentration. So, Kq into Q I have written in that term, and then the fluorophore is getting quenched, right, because of the creation of the extra non-radiative pathway in its photophysics. Clear? Now, question is that what will happen if the viscosity of the medium will increase? If the viscosity of the medium will increase, then those quencher molecule will not be able to interact with the fluorophore in that way. That means, the quenching phenomena will not be that strong, right. In other words, this whole phenomena can be represented as that this is kind of a dynamic process, like this is a collision between the two molecule is responsible for such kind of quenching. The quencher molecule should come and interact with the fluorophore, then only such kind of process will take place. So, this is a dynamic process. So, this type of quenching is known as dynamic quenching, right. So, this type of quenching is known as dynamic quenching. So, this is resulted from the collisional encounter. That is what I said right now, right, between the fluorophore and the quencher. So, the fluorophore and quencher interaction, like this fluorophore and quencher interaction should be takes place, right, within the lifetime of the fluorophore. This is also an interesting thing. Okay. So, this is another time this is also called as the collisional quenching, and this is, you please note that this is diffusion controlled. Okay, now, let me discuss another type of phenomena. Suppose you have a molecule F which is in the excited state, right, will give you light, right. So, F plus H nu emission, and you have started with some quantity of F, right, and when you excite it, you will create this F star, and then F star will form the F, because of it has a radiative process, it also has a non-radiative process. So, depending on that you will get this H nu emission. Now, what if when you are adding the quencher here, so addition of quencher will take away some fraction of the F and complex with it, so that you are unable to excite that fraction of F from the ground state to the excited state. What I wanted to say is that now some FQ complex which you cannot excite to the F star state, that means the concentration of the F star state will decrease. If the concentration of the F star state will decrease, that means the fluorescence intensity will decrease. If you increase the quencher concentration Q, then more and more F will get complexed with Q, right. So, this is a ground state phenomena. So, here is the ground state complex formation in this case, is not it? Now, that I can also equate it. Let's designate the fluorescence intensity as is proportional to the concentration, right. So, I can write the fluorescence intensity is proportional to the concentration. So, let us say the fluorescence intensity is F star 0 when there was no quencher added. That means, whatever the F star you can create from the ground state by your suitable excitation source, that is the concentration, which is obviously proportional to the fluorescence intensity of this sample. So, I can just simply take F star 0 is my initial fluorescence intensity without addition of the quencher molecule. Now, once you will add this quencher, right. So, fluorescence intensity will be different than this. So, let us write that F star is the fluorescence intensity in presence of quencher, then I can write F star equal to F star 0 minus something minus F Q. So, some fraction of the fluorophore is getting complexed with Q, which I cannot excite anymore, right, and all the remaining F are excited to the excited state, or I can write F Q, that concentration of F Q equal to F star 0 minus F star. So, I can write some equilibrium constant F Q divided by F star into Q. So, this is equal to F star 0 minus F star divided by F star into Q. This is equal to 1 over Q into F star 0 by F star minus 1. So, rearranging F star 0 by F star is equal to 1 plus Ks into Q, and this F star 0 by F star is nothing but I 0 by I, that is what I argued like. So, I can write I 0 by I is equal to 1 plus Ks into Q. You see this form of this equation and you look at the form of the other equation. So, you see this form phi 0 by phi, this form phi 0 by phi equal to 1 plus Kd into Q, here I 0 by is equal to 1 plus Ks into Q, they are exactly same. So, in this case also, right, this case means where the ground state complex formation takes place, and because of that the fluorescence intensity is decreasing as I am adding the quencher molecule in my medium. So, in this case the mechanism is completely different. In the other case the mechanism was the quencher molecule is coming and interacting with the excited state of the species which is responsible for the fluorescence, and it is creating another pathway, another non-radiative channel, so that the fluorescence intensity of the molecule is decreasing, because fluorescence intensity means quantum field, that means Kr by Kr plus all the non-radiative processes. But in this case there is a ground state complex formation is taking place. As this ground state complex formation is taking place, I am not going to have enough fluorophore in the excited state, and that is why the fluorescence intensity is decreasing. The two phenomena are completely different, the mechanism is completely different, but the equation are very similar or exactly same, right. So, in this case also, and so this type of quenching is known as static quenching. I will write here somewhere here. So, static quenching. So, in this case also, if you plot the fluorescence intensity or the ratio I 0 by I versus concentration of Q, what you will going to see? You will also going to see a straight line with intercept 1, exactly same, right. Now, question is how we are going to distinguish whether it is a static quenching or it is a dynamic quenching. Here you see I am exciting the molecule in the excited state, the nature of the excited state remains same, only some part of the molecules cannot be excited to the excited state because of the ground state complex formation, but the nature of the excited state remains same. Same to what? Before addition of the quencher, same to before addition of the quencher. So, there is no change in the excited state property, that means whatever the value of Kr and whatever the other values of Kn they remain same, that means there will be no change in the lifetime. Please note that when I am going to plot tau 0 by tau in similar fashion, what you will going to see? Always the ratio is 1, even you increase the quencher concentration, is because lifetime does not depend on the concentration of the species, or lifetime is the property of the excited state, and in case of static quenching the ground state complex formation is taking place, which is just removing a fraction of the fluorophore from my observation, right. So, excited state remains same. So, I can readily distinguish whether it is static quenching or dynamic quenching by measuring intensity as a function of quenching as well as by measuring the lifetime as a function of quencher, right. So, in this case you see the measurement of the lifetime is so important. Only using this lifetime data you will be able to tell whether this quenching is static quenching or is a dynamic quenching. Okay. Okay, let us finish here and we will continue our discussion on the next lecture. Thank you very much.